Hi there, my name is Stuart Milligan and I'm going to tell you about the central control of reproduction in a series of shortish lectures. The idea is not to fill you full of facts with these, but to give you a feeling for the whole subject area, about where it's come from, where it may be going, and more specifically to try and get you interested in reading more around the subject. From the early days of physiology, the reproductive system has provided a very important model. Why? Because unlike almost every other system in the body, like the kidneys, the heart, the liver, it's not essential to life. It's also experimentally easily accessible. The testes are way down in the scrotum, the ovaries are in the abdomen, and the changes in the reproductive tract are very dramatic. Huge changes take place, for instance, at puberty. So experimentally, it's a very good system to look at. Here we see the testes in a prepubertal mouse, very small. These are the seminal vesicles. At puberty, the testes grow rapidly. Here they are. They secrete a lot of testosterone, and the testosterone has multiple effects around the body. For instance, stimulating the seminal vesicles. These can be very easily weighed. So this is an indirect marker of testosterone. And testosterone is an indicator of what's happening within the testis. The testosterone doesn't just affect the secondary sexual organs, like the seminal vesicles, but also affect other things in the body, including things like behavior. And many of the early studies of how the brain works and how behavior is modified, especially the hormonal control of behavior, came from looking at sexual and aggressive behavior, driven by things like testosterone. So a very important basic model for looking at endocrine systems, how endocrine systems affect the brain, and how the reproductive system works per se. Exactly the same kind of thing can be done in the female system. Here we have a prepubertal mouse, where these little string-like things are actually the immature uterus. Here are the tiny ovaries. At puberty, this whole thing is stimulated. The uterus changes under the influence of estrogens to this great big thick juicy tissue. Here are the ovaries, and this is mainly due to estrogens coming out of the ovary driving the uterus. But as before, estradiol has many other, or estrogens in general, have many other effects within the body, affecting many other tissues, including the brain and behavior. Our basic understanding of the general system was really established in the first part of the 20th century. In the early 1930s, FSH and LH coming from the anterior pituitary were isolated, and at about the same time, the steroids, testosterone and estrone and estradiol were isolated as well. And the general idea was that the pituitary drove the ovary and testis, which produced eggs and sperm, as well as steroids, and these steroids could feed back onto the anterior pituitary to regulate the drive from FSH and LH. So negative feedback was seen as a dominant control feature. But it was obvious other things must be going on as well. There was continuous sperm production in males, and that strongly contrasted with the ovarian cycles in females, with menstrual cycles in primates and estrous cycles in most other mammals. And these had to be fitted into the much bigger picture of the life cycle. Here we have reproductive potential on the vertical axis. Along here we have age in humans. Reproductive potential, or reproductive system, is essentially not functioning until puberty. Then it suddenly switches on, continues at a high level in both males and females, but then at the menopause it switches off in females, whereas males go on throughout life producing sperm. So how does the simple picture of the pituitary driving the gonadal system fit with this contrasting sperm production continuous in males, estrous cycles in females, menstrual cycles in females, and this turning on at puberty and ending at the menopause? Something else must be going on. And then there was the issue of breeding seasons. It was obvious whether you looked at birds or mammals in temperate regions that discrete breeding seasons fitting in with the time of year took place. Here we have the quail, which essentially breeds in the summer. So it starts mating in the spring, egg laying, incubation, and the whole idea is the production of their offspring is synchronized with the external environment when there's maximal food supply, optimal conditions to maximum survival of the offspring. These were called, birds like this, long day breeders. Why? Because they tend to breed in the long days of summer. Things like sheep were called short day breeders. Why? Because they started mating in the autumn when days were shortening. They then became pregnant, and pregnancy in the sheep is just over 150 days, so they could deliver in the spring, again maximizing the conditions for optimal survival of the offspring. So what could explain breeding seasons? Somehow reproduction was tied into the outside world. And the seasonal timing of breeding was very precise, because it wasn't just breeding itself, it was all the other things associated with breeding. Here's a good example: the growth of antlers in red deer. They're shed. Here we are, a single red deer stag over 3 seasons, April the 3rd 1 year, April the 2nd another year, March the 30th another year. We'll pick up seasonal breeding a bit later, but the fact that external factors such as seasonal cues could control this system indicates that some other inputs must be occurring into the pituitary control of the ovary and testis. And farmers knew that there was fine-tuning of reproduction as well. If you introduce a ram into a flock of ewes, it accelerates their breeding season, brings on the breeding season earlier. And we now know it's to do with pheromones coming from the ram, inducing that reproductive change in the females. But the social environment was also impacting the system. So external factors, seasonal cues, social cues, all of these inputting somehow to the anterior pituitary, driving the cycles in the ovary and testis. And there was one other observation that was to prove pivotal in unravelling how these external factors could control the anterior pituitary. It was due to observations about what happened in rabbits. Now, what happens in rabbits when they mate is that the female rabbit ovulates about 12 hours later. So a very specific stimulus and a very specific event within the ovary. And I'm now going to take you through just how mating in rabbits, inducing ovulation, actually helped unravel this whole pathway. It's real classical physiology, all done in the 1920s onwards, but culminating in our current understanding of the intricate control of the brain over the reproductive system. Let's go back to 1929. The first of a series of papers was published, this one by Morris Friedman, The Mechanism of Ovulation in the Rabbit. 1. The demonstration of a humoral, that is hormonal mechanism. I'm going to read through most of the introduction because it really does describe what was known and what wasn't known at the time and the kind of theories and thought processes which were going on. It starts off: the rabbit differs from other laboratory rodents in that it does not ovulate spontaneously, but only after coitus. Neither artificial insemination nor injections of sperm or semen are successful in provoking ovulation. So this is an important statement because they didn't know why coitus was inducing ovulation, and it could have just been this, or could have just been, it could have been that the presence of semen in the female reproductive tract was the essential stimulus. But that didn't work. Yet ovulation regularly follows coitus with a vasectomized buck. A vasectomized buck, so he wasn't inseminating any semen because he'd been vasectomized, and can be produced by rubbing the vulva of a female in heat. From such considerations, it is clear that if the male secretions contribute anything at all to the conditions requisite for the initiation of the process of ovulation, they are not of great importance. Marshall, who was a great reproductive biologist who published a very important series of books on reproductive biology, Marshall holds the view that the phenomenon is reflex in nature. In his opinion, the fact that ovulation only occurs after coitus, and then at a definite time, 10 hours afterwards, points to the conclusion that the follicles discharge in response to a stimulus conveyed to the ovary by its nerves. So he was saying that mating somehow by the nerves going to the ovary, that was a stimulus for ovulation. But Friedman says that a reflex should require 10 hours for its completion seemed improbable. So this reflex idea that people weren't going with. Nevertheless, there's been to date no decisive work to confirm or refute this hypothesis, so he undertook some experiments. So what did he do? He transplanted the ovary somewhere else. If you transplant the ovary somewhere else, it loses its innervation. And do the rabbits still ovulate? Yes, they do. So it can't be the nerves. It must be, what he set out to demonstrate, a humoral mechanism. So Friedman went on to the next experiment. Mechanism of ovulation in the rabbit. Number 2, ovulation produced by the injection of urine from pregnant women. Okay, let's have a look at the introduction of this one. And he starts off, from the demonstration that ovulation may occur in the transplanted ovary of a rabbit, it is obvious that the ovarian nerves are not indispensable to the process. So he's saying the Harris theory of the nerves being the essential thing going to the ovary, causing reflex ovulation, can't be true. And it is equally obvious that there is present some humoral mechanism. The question arises, however, of the significance of this humoral mechanism when the nerve supply to the ovary is intact. If it is of great significance, one should be able to provoke ovulation in the unmated rabbit by reproducing these humoral changes. And that's what he went to do. But why the urine from pregnant women? Well, what he was doing was following on from this observation. Pregnant women, we know in their urine now, they produce a lot of HCG. And HCG has got an activity very similar to that of LH because the structure of HCG is very similar to LH. And it had recently been demonstrated, that is in 1925, that if you inject the urine of pregnant ladies into an immature mouse, that will cause follicle growth and ovulation. So there was something here they knew which would cause follicle growth and ovulation in mice. And what Friedman did is say, okay, does it work in rabbits? Yes. So he could induce ovulation in rabbits just by injecting in urine from pregnant women. Some hormone here was causing ovulation. The observations of Friedman were followed up in England by a series of studies, also entitled Studies on Ovulation. 1. The relationship of the anterior pituitary body to ovulation in the rabbit. So, given that the idea was there may be something hormonal causing ovulation, and given that they knew that the anterior pituitary gland produced very many hormones important throughout the whole body, including for stimulating the ovary and the testes, although FSH and LH hadn't yet been isolated, they said, okay, what happens if we remove the anterior pituitary? If we remove the anterior pituitary, will ovulation still take place? So they did this experiment. They mated the rabbit. They removed its pituitary within 1 hour of mating. Removing the pituitary is called hypophysectomy, and they looked to see what would happen. And sure enough, no ovulation. Removing the pituitary blocked ovulation. So it looked as though there was something coming from the anterior pituitary which was mediating the response of mating and ovulation. Following on from that, more studies. This one is number 4 now in the series. Induction of ovulation in the hypophysectomized rabbit by administration of anterior pituitary lobe extracts. So what they did, that given that hypophysectomy within 1 hour of copulation inhibits ovulation, they said to demonstrate conclusively that a genuine hormone effect was involved, it was necessary to show that the pituitary body could be replaced by an extract. So what they did was inject in an extract of the anterior pituitary gland and showed that could also induce ovulation. So the idea was beginning to develop that the anterior pituitary was central to the control of reproduction. The anterior pituitary produced hormones which could control the ovary and testis and induce ovulation, but the anterior pituitary and reproduction was somehow being controlled by external cues, like seasonal cues, social cues, and in rabbits, mating. How could those external cues get into the anterior pituitary? Well, the external cues were obviously sensory cues, so the idea was the brain might be involved. So to investigate that, Marshall came up with the very easy idea: what about electrical stimulation? Could we electrically stimulate the brain and simulate some of the effects of the external controls? And that's what he did. Electrical stimulation, a broad brush electrical stimulation of the rabbit brain. What happened? That could mimic mating and induce ovulation. So it looked as though we were on the way into investigating how the brain might control the anterior pituitary. It was known that the anterior pituitary sat just below the hypothalamus, a very old area of the brain, essentially in the integration of many things: appetite, fear, thirst, temperature regulation, metabolism. So instead of broad brush electrical stimulation of the whole brain, what about limiting the electrical stimulation to the hypothalamus, and even then to particular areas of the hypothalamus? So electrically stimulate the hypothalamus. What happens? Yep, ovulation. So it looked as though hypothalamic control of the pituitary might be a link in the chain. This was an important part of the control. Electrical stimulation, not of the hypothalamus this time, but of the pituitary itself. Perhaps that was responding to the electrical stimuli directly. Nope, electrical stimulation of the anterior pituitary, no ovulation. Stimulation of the hypothalamus, then ovulation would occur. So the hypothalamus seemed to be a key link in the chain. And I'll now quote from a review looking back on these historical observations. It says, "The paradox is that the anterior pituitary, which is crucially involved in the response to environmental, seasonal and mating stimuli, is clearly under control of the CNS. But despite considerable efforts to find one, it has scant or no innovation." Harris wrote, "The pars distalis of the pituitary may be described as a gland under nervous control, but lacking a nerve supply. How then is the nervous system able to control the release of the anterior pituitary hormones?" Let's look at the thing in real life. This is the pituitary gland. We're looking at the underneath side of the rat brain, and the pituitary gland sits just underneath the hypothalamus, which is all in here. So this is the anterior pituitary, and these are vessels which are coming down, linking the hypothalamus with the pituitary. Here we're looking at the same thing, but with everything a bit more exposed. And this is the pituitary, this is the hypothalamus, and the blood vessels now have been injected with Indian ink to really make them show up. So that anything black is a blood vessel. So we can see lots of blood vessels linking the hypothalamus and the pituitary. These were called the portal blood vessels because they seem to break up in the pituitary and they seem to be in capillaries in the hypothalamus. So a portal blood system carrying blood between the hypothalamus and pituitary. The idea was beginning to develop in the mind of Geoffrey Harris, who was working in England, that the hypothalamus might be producing something which was sending messages down these portal blood vessels to the pituitary. But that required the blood vessels to be flowing in that direction. The portal blood vessels should be carrying the blood from the hypothalamus to the pituitary. Now, the trouble is that the current idea was that in fact the blood was flowing the other way. But that idea was just based on anatomical studies with no evidence at all. Then a French worker in 1935 said in a toad, he'd looked at it, in a living toad, anaesthetized, and the blood was flowing from the hypothalamus down towards the pituitary. And then a bit later, some years later, in the rat, Harris looked at it directly, looked at these blood vessels under a microscope, and sure enough, the blood was flowing in that direction, from the hypothalamus down towards the pituitary. Harris began to develop the idea that nerves up in the hypothalamus might release things into the portal blood vessel, and those factors could control the pituitary. This was based partly on the observation that nerve cell terminals occurred up here in close association with the portal blood vessels. Those nerve cell terminals seemed to contain lots of little vesicles, ideal for neurosecretion. Now, the whole concept of neurosecretion at this time was very new, so even that idea didn't go down very well. But his idea was neurosecretory products might go down the portal blood vessels and control the various pituitary hormones. Harris pursued his ideas throughout the 1940s and early 1950s with a very elegant series of experiments. Things like this, asking how important are these blood vessels, what can you do? Well, what you can do is cut the portal blood vessels. If you cut the portal blood vessels, surely you would disrupt the signalling between the hypothalamus and pituitary. Well, that was the idea, and it should have worked, except it didn't. It seemed to have no effect at all. So if cutting portal blood vessels didn't have an effect, surely that destroys this idea that factors coming from the hypothalamus to the pituitary were important in controlling the pituitary itself. But Harris realized there was a flaw in this experiment. Simply cutting the portal blood vessels wouldn't actually disrupt the blood supply for very long because they simply regrow. So perhaps it wasn't surprising that simply cutting the portal blood vessels, there was no major disruption to the rest of the endocrine system because the portal blood vessels regrew, the factors could come down again. So he had to do something else. What could he do? He thought, let's stop the portal blood vessels regrowing. And we can do that by inserting a very thin impervious plate across here. And what he used was a little piece of waxed paper. Cut the portal blood vessels, insert the wax paper, the portal blood vessels couldn't regrow, and what happened? Pituitary function largely ceased. Effects were seen, for instance, on the testis regressed. All the other anterior pituitary functions were disrupted, so these portal blood vessels seem to be very important. But it would be easy to argue that the decline of pituitary function was simply because you disrupted blood vessel supply to the pituitary. So how could you control for that? Well, the simple thing it was to transplant the pituitary somewhere else in the body where it would still get a blood supply, but wouldn't have the portal blood vessels. And that's what he did. And it's easy to transplant the pituitary. You can put it underneath the kidney capsule. It gets a revascularization and pituitary function is disrupted again. For instance, the testis shrinks. So it looks as though these portal blood vessels are somehow very important. What we've seen is how science progresses. It was a series of observations, thought out, well-designed experiments, and interpretation of those experiments leading on to the next one. And in 1948, Harris began to put all his ideas together, and he published a paper on the neurovascular control of the anterior pituitary gland. And his idea was that the hypothalamic nerves up here produced hormones. He called them releasing hormones or releasing factors. They travelled down, they were released at the nerve cell terminals in the median eminence, which is essentially the bottom bit of the hypothalamus, well vascularized by the portal blood vessels. Those releasing factors or releasing hormones went down the portal blood vessels into the anterior pituitary, where they controlled the release of each of the anterior pituitary hormones. What we've seen is how science progresses. It was a series of observations, thought out, well-designed experiments, and interpretation of those experiments leading on to the next one. And in 1948, Harris began to put all his ideas together, and he published a paper on the neurovascular control of the anterior pituitary gland. And his idea was that the hypothalamic nerves up here produced hormones. He called them releasing hormones or releasing factors. They travelled down, they were released at the nerve cell terminals in the median eminence, which is essentially the bottom bit of the hypothalamus, well vascularized by the portal blood vessels. Those releasing factors or releasing hormones went down the portal blood vessels into the anterior pituitary, where they controlled the release of each of the anterior pituitary hormones. And so the next question was going to be, if you've got hypothalamic hormones here coming down, and we knew that by this time that the pituitary produced a number of very important anterior pituitary hormones: FSH, LH, ACTH, growth hormone, prolactin. There must be different hypothalamic hormones controlling the release of each of those anterior pituitary hormones. So the next question was, what were these hormones? What were they like? What were the nerves which were producing them? The search for the hypothalamic hormones resulted in the Nobel Prize for Medicine or Physiology in 1977 being awarded to these two jointly, Roger Gilman and Andrew Shalley, for the isolation of the first hypothalamic releasing hormones. The first one came in 1969, which was TRH, and then in 1971 gonadotrophin releasing hormone, GnRH. While they both share the Nobel Prize, their search for these releasing factors or releasing hormones was not the most auspicious tale in science, because although it involved a lot of work, these two, although they initially worked together, fell out, and there was intense rivalry between the two, a competition between the two to get there first. But it's also to note that the Nobel Prize for Medicine and Physiology, or Physiology, in 1977 was shared also with Rosalind Yalow for an entirely different reason. She was actually the founder of one of the major revolutions in the whole of endocrinology, the ability to measure small quantities of hormones in the blood by radioimmunoassay. Until this point, most measurement of hormones was done by some kind of bioassay, using a biological response to measure the tiny quantities of hormones in the blood. Now the trouble is, the amount of hormones in the blood were actually so tiny that it really wasn't possible to do many things with them. You either needed a big source of the hormone, like the original gland, or you needed to collect vast amounts of the stuff by taking a lot of blood or repeated samples. The reason being is because until that point, there was no specific chemical reagent which would specifically bind with any of the hormones anyone was interested in. But Rosalind Yalow, back in the 1950s, they were working with insulin, and they saw that patients injected with animal insulin showed reduced clearance of insulin from the blood. And they wondered why this should be the case. Why should you have reduced clearance of insulin from the blood? And they said, maybe we've got antibodies there. So, and sure enough, they found there were antibodies to insulin. Now, if you think about it, antibodies are a very specific chemical which will bind to the hormone you're interested in. So they said, we've now got a very specific chemical. Those antibodies will only react to insulin. And she said, okay, what we could do now is to develop those very specific chemicals into some kind of assay system. And the assay system was to do with competition of endogenous insulin with radio-labeled insulin for the antibodies. Depending on how much endogenous insulin you've got, the radio label will bind to a greater or lesser extent, and that will give some indication of how much endogenous insulin there really is. So she used these antibodies as a way of measuring insulin in the blood, and that was the foundation of radioimmunoassays. That's why she got the Nobel Prize.